All right, it's a great pleasure to introduce my student, Li Tang, as a next speaker for this, his talk, Precisely Size Control Drug Silica Nanoconjugate for Cancer Therapy. So Li is a fifth-year graduate student in my group, and he graduated from Peking University in 2007, right, and then joined my group in fall semester of 2007. He has been working on multiple projects. About 2.5 years ago, or 3 years ago, he started this project, and independently, he came up with this idea to graft drug through the soldier chemistries. You're going to see the really beautiful size control that he can do. So I'm not going to talk too much about his science. I'm actually going to tell you a little bit about this program and how this program, how he can benefit from this program. How he has benefited from the program for his trainings. So he has been working on the cancer and nanotechnology. Okay, so cancer and nanotechnology, you're dealing with cancer, which is very biology or clinical-based science, and material science chemistries. Okay, there's a huge boundaries between those two distinct fields. There got to be someone to step into these gray areas to really crosstalk, to make the connections of all the physicians and the chemists and material scientists. Okay, so there got to be someone. Doctors or clinicians, they're way too busy to do this, right? They're not gonna, never gonna learn this chemistry or material science. But it has to be someone like Li to step in these areas and to do a lot of this clinical type of work and understand different areas. So I think that's really what this the M-CNTC program provided to the students like Li. Okay, so Li has been doing an exceptionally good job in these areas, and he has been able to push and chasing those very busy people get what he wants. Okay, and without Li, I can never imagine we can establish so many very successful collaboration and to the level we are at right now. So he's in my group working on this silicon nanoparticle development. That's his core technology. And he has been driving this areas very deep. And he's co-advised by Tim Fan. And what he learned from Tim is the injection technique and many of these animal techniques. So Li has been personally involved in many of this, doing a lot of the in vivo study himself, doing tail vein injection, tissue harvestings. You know, he's been keep Tim Fan busy, getting all this metabolic cage. Is that right? To do the radioactive following this phases. You know, something I never heard of. So Li has been keeping those people busy to get some really fancy study completed to do a complete evaluation of his silicon nanoparticle. And then he pushed to the new frontiers working with Bill Hanfridge, established collaborations and getting a lot of this MCF7 models and 41 cell models for this metastatic cancer models. You're going to hear some of the stories he developed these collaborations. He also working with Yilu lab, working on this liposome project. He does not have time to talk about this project. And very impressively, he pushed for the developing new collaboration with John and with Kazan Bogan and also Dr. Dabrowski for this PET imaging. I believe my lab is probably one of the most active labs using this PET imaging facilities, thanks to Li's efforts. And beyond this M-CNTC program, he also developed some new collaborations with Reza Abdi at Harvard Medical School, applying the new use of nanotechnology for the different areas. So not only the science, research, but also the training of Li in many different aspects. For example, the interaction with various people and the service, community service, and outreach activities. Okay. So he has been involved, presented for his own research and also on behalf of M-CNTC at UC San Diego, Golden Conference, and also the NCI Alliance in Boston. And also he has served as a member of the student leader councils, inviting speakers. So he basically functioned kind of like junior faculty levels. Maybe in a few years, he will have more experience. And he also learn and teach in the Bio-Nanotechnology Summer Institute. So this takes a lot of time to prepare. It takes about a week of time. But he gained and learned more than what he can learn by spending a week in the lab, honestly. So this is a really great module to teach the students in many different aspects. Of course, there'll be outreach activities. I don't have time to emphasize. So basically, it's a great platform for the training of the students in this cancer nanotechnology area, not only for the collaborations, multidisciplinary research, and also multitask and a variety of different training as a young scientist. That's what we need for this cancer nanotechnology areas. Okay, so here's Lee. He will talk about his silicon nanoconjugate.
Okay. Thank you for the introduction. So I will talk about our research results in this project. So basically, the focus of our research is developing novel nanomedicines. Nanomedicine, which utilize nanotechnology for the biomedical application, has been developed in the last 30, 40 years. Nanomedicine may offer some new opportunities and hope for both diagnosis and the therapy of cancer because they have some advantages over the conventional chemotherapeutics. For example, using nanocarriers, you can protect the drug from degradation in the physiological conditions, and it can design to passively target the tumor through the so-called enhanced permeability and the retention effect, EPI effect, and also can be designed to control drug release to minimize the side effect. Some of the successful examples of nanomedicine, for example, doxil and abraxane, these are already in market for years. So although nanomedicine is very promising for the cancer therapy, there are still quite a few issues. For example, the overall accumulation of the nanomedicine in tumor is still pretty low, usually like less than 5%. And also the penetration in the tumor tissue is very limited. It prevents the drug to kill the cancer cells deep in the tissue. And the unclear nanotoxicities further prevent the application of nanomedicine in the real applications in human. So to address all of these issues, let's first take a look at how the nanomedicine works in the animals or in a human patient. So by injecting the nanoparticles into the bloodstream of a mouse, there are at least 3 biological barriers for nanomedicine before it can reach the cancer cells to really kill the cancer cell. So first one is the systemic barriers, how the nanoparticle to really target the tumor tissues. Some early studies showing the leaky vasculature structure of the blood vessels in the tumors. So nanoparticle with a certain size can take advantage of these pores to penetrate these pores and accumulate in the tumors. So clearly, it will be very important to optimize the particle size to fully take advantage of this effect to target the tumors. The second issue is the tumor penetration. Because of the high interstitial pressure in the tumor tissues, many of the chemotherapeutics nanoparticles can only penetrate very limited and cannot kill the cancer cells distant from the blood vessels. So clearly, it will be important to tailor the particle size to improve tumor penetrations. Another barrier is the cellular internalizations. So some study already showing that this process also highly size dependent. It will be important to optimize the intracellular delivery by optimized particle size. So to overcome these barriers, the strategy in our lab is trying to precisely control the physical and the chemical properties of nanomedicine, especially size, which we think is one of the most important parameters of the nanomedicine. So our hypothesis is there is an optimum size or size range of nanomedicine to reach the most efficient cancer therapies. So to test this hypothesis, we want to first design such a drug delivery system. First, we want to precisely control the particle size, and which will allow us to study the size effect of nanomedicine in different biological systems. The second, we want to develop it as a real drug delivery system, meaning that we can incorporate the real anti-cancer drug and potentially for the clinic translation. So we were thinking about the silicon nanoparticles because it can be excellently controlled over particle size. And silica is generally considered as a bio-inert materials. Now the question is how to utilize silica nanoparticle as a drug delivery vehicle. So we were finding some interesting aspect of the silane chemistries. We noticed that for typical silane compound has 4 of these silicate acid bonds. They can hydrolyze, condense with each other with forming the silica. So by controlling the conditions using the Stolper method, we can obtain the nanoparticles with precisely controlled size. But if we save 3 of the silicate bond and leave the other one to conjugate with some functional molecules, for example, chemotherapeutics, then we can synthesize this drug containing silane compound. And this compound can go through the same reaction and incorporate easily into the size-controlled silicon nanoparticles. So to test these ideas, we first use a pyrene as a model drug. And we can, as shown in these SEM images, we can really successfully formulate these pyrene conjugated silicon nanoparticles ranging from 200 to 20 nanometer. And you can see the size are extremely monodisperse, well-controlled, and highly reproducible. So besides pyrene, we can also follow this strategy. We can synthesize many of these compounds containing the silane groups. For example, camptothecin, which is an anti-cancer drug, paclitaxel, docetaxel, and also we can synthesize the fluorescence dyes to tracing nanoparticles in vitro and in vivo. And we can modify the surface by using a polymer, for example, polyethylene glycol containing the silane group. And we can even synthesize this silane bridged compound with a responsibly degradable domain in between. By integrate this component into the particles, we can tune in the degradation of the whole particles. So besides the size control, we can control many properties of this nanomedicine by control the chemical and the molecules we are using. For example, we can control the formulation very well. These are very important properties by developing a nanomedicine for potential clinical translations. For example, we can achieve very high drug loadings using a CBT, camptothecin conjugated silicon nanoparticles. We can get higher than 20% drug loading and easily get more than 80% incorporation efficiencies. We can scale up to gram scale, and we can get solid formulations by lyophilization and redisperse, which is ready for the injection and maintain the original size. And importantly, we can, by controlling the chemistry, to control the release profile of the drug from the silicon nanoparticles. For example, by using different linkers between the drug and the silicon nanoparticles, we can tune in the release kinetics from more slower sustained release to a much faster release. So it depends on the application, we can tune in the different linkers for the drug conjugation. So after we get such a precisely size-controlled drug containing silicon nanoparticles, the first question we want to answer is, does the size really matter for the overall efficacy of the tumor therapies? So to test this size effect, we designed the in vivo tumor reduction studies by implant the tumor to the nude mice. Then we give the treatment of different groups. Then we monitor the tumor growth over time to evaluate the inhibition effect of these different treatment in terms of the tumor therapies. We evaluate these 6 different groups, PBS buffer as a negative control, and the blank silicon nanoparticles to out any possibility from the delivery vehicle itself, and also irinotecan as a clinic drug as a kind of positive control. And for our CPT containing silicon nanoparticles, we evaluate these 3 different sizes, 250 and 20. So basically, from the result, the tumor growth pattern, you can clearly see the PBS group and the blank silicon nanoparticle group. They show the tumor is grow very aggressively, and the clinic drug irinotecan showed pretty good inhibition of tumor growth. However, it is highly toxic to the mice, which can be indicated by the dramatically body weight drop after treatment. So our nanoparticles has minimized this side effect and still show the inhibition of the tumor growth. So based on the growth pattern, you can see the 50 nanometer particles slightly better than the 20, and both the smaller particles that are much better than the 200 nanometer particles in terms of the inhibit tumor growth. And we performed the statistical analysis, and truly the smaller particles show the significantly improved efficacy of the in terms of inhibit tumor growth.
What kind of tumor was that?
This one is the M-CNTC, the human breast cancer tumor.
You injected on the back.
Yeah, the tumor is a subcutaneous injected into the flank of the mice, and the drug is injected from the tailwind injections. So clearly the size really matters for the overall efficacies. Now we are trying to understand why the size is so important. So first, we performed the biodistribution studies using the radio-labeled silicon nanoparticles of different size. So these are copper-64 labeled silicon nanoparticles we inject into the tumor-bearing mice. And the gray map is indicated from the CT imaging to indicate the position of the mice and the position of the tumor, which is indicated by the yellow circles. The color map is from the radioactivities from the radio-labeled nanoparticles. So clearly you can see from the imaging that the 50 nanometer particles show the highest accumulation in the tumors. And then is the 20, and these two are much higher than the 200. So these results further confirmed by quantify the radioactivities using the gamma counter. And we did see that 20 and 50 nanometer particles show almost about twofold accumulation. Penetration in the tumors. So after they reach the particles reach the tumor tissues, then how efficiently they can deliver the drug really inside the cells. Then we evaluate the cellular internalizations of different size particles. So here we compare the uptake of three different size particles in the HeLa cells, and the particles are also labeled by the rhodamine as shown in the red color here. The nucleus of the cells is stained by DAPI as shown in the blue color. So clearly you can see the smaller particles showed enhanced uptake by the cancer cells. And we also quantified the cell uptake using flow cytometry to study the kinetics. And the smaller particles also showed much faster internalization into the cancer cells. And interestingly, we found that different size particles, they also go through a dramatically different pathways into the endocytosis into the cells. So by performing some inhibitor studies. So besides the systemic distributions, we also study the size-dependent lymphatic distributions. So the purpose here is we want to develop some efficient imaging probe for diagnosis of the tumor metastasis. Then we want to evaluate if by controlling the particle size, if we can achieve the enhanced accumulation in the lymph nodes. So in these experiments, we give two different size particles through Hawk injections. Then we want to use a PET-CT imaging system, see like how efficiently they can accumulate in the most close lymph nodes. So here, as you can see, we give 20 and 200 nanometer particles. 10 minutes after injection, the 20 nanometer particles start to accumulate in the sciatic lymph nodes, as shown in this PET-CT imaging. And after 6 hours, they are still they reach the highest radioactivities, and these activities can maintain even to 22 hours. So, but 200 doesn't show very obvious accumulations. You can probably easier tell the accumulation of the 20 nanometer particles accumulation in the lymph nodes. So these results indicate that by really control the particle size, we can achieve much efficient lymphatic accumulations. So another important application of nanomedicine is used to inhibit and prevent the tumor metastasis. So we are testing if using our size-controlled silicon nanoparticles can prevent the tumor metastasis. So here we're using the 41 cells, which is a highly metastatic tumor model, by injecting through the tailwind. And after several days, these luciferase engineered 41 cells, they can quickly develop the tumor metastasis in the lung area of the mice. Then we give 3 different treatments of give 3 injections of different treatment. Then we can measure the luminescence from these cancer cells to evaluate how much metastatic tumor has been developed in the lung area. So you can clearly tell by this imaging, by giving the CPT containing silicon nanoparticles of 50 nanometers, we can see the significantly reduced tumor metastasis developed in the lung areas. And we further confirm the result by taking out the lung and performing the pathology analysis. And we did find the dramatically less of the tumor nodules developed in the normal lung areas. And we can quantify the luminescence, and we did see the significantly reduced of the luminescent signals. And by performing the biodistribution using PET-CT imaging, we did find 50 nanometer particles show the enhanced accumulation in these metastatic lung areas. So as I said, we want to develop these silica-based nano medicines as a clinical translational medicines. So the issue we have to address is the long-term toxicity or biocompatibilities. So we to study the long-term safety of these silicon nanoparticles, we give a large dose injection of the blank silicon nanoparticles into the mice. Then we monitor these mice up to 4 weeks. During this time, we monitor the behavior of the mice and measure the completely blood chemistry and the CBC panel of these mice. So basically, I can only show some of the data here, but basically we didn't see any abnormal change.
Cells. So it looked like they were on the side of the animal. Did you do them in the fat pad?
We didn't do the fat pack. We inject 3 tumors on the flank of the mice, each mice.
Yeah.
So they 2 on the front and 2 at the back. So they are subcutaneous injection.
Oh.
They are developing the xenograft tumor models for us to evaluate the inhibition of tumor growth.
So you're not really using it as a breast cancer model then? You're just
So
using
we are using as a xenograft model, not the orthotopic tumor models.
Yeah.
Right.
So the fact that the smaller and smaller particles would have better and better in distribution and potential uptake, would it not something that you would expect? The bigger particles would have less diffusion, would have having difficulty in penetrating, so that would be obvious, right? What is not clear to me is that how does it get distributed. You are injecting in bloodstream or in a tissue, right?
Yes. So so I think in terms
In
of
the bloodstream, right?
into the bloodstream of the
Bloodstream.
animals,
So
right?
How does it get out of the bloodstream into the tissue?
Yeah.
That's the part that's confusing to me.
Yeah. So all of these particles that have been evaluated show here is without active targeting ligand. So just based on the passive targeting. So many of the literatures, they reported the so-called EPR effect, enhanced permission and the retention effect. That's because of the leaky vasculature structure in the tumor vessels. So because of this leaky vasculature, particle with a certain size, they can penetrate into these tumor tissues. And without a very functional lymph nodes, lymphatic vessels, they cannot get out. So based on this enhanced permission and retention effect, particle with a certain size, they can really passively target tumors when you inject into the bloodstream. So I think based on the passive penetration, as I showed in the ex vivo experiments, it is true that when you control the particle smaller, they can really passively penetrate deeper. But the question is, in a live animal system, is if you are penetrated too much, you may just buy through the whole tumor tissues, and you cannot really accumulate these particles in the tumor tissues. So there got to be an optimal size of the nanomedicine to really maximize the accumulation and achieve the most effective therapeutic efficacies. So that's why we want to use a system which can be really precisely controlled over size, which cannot be achieved by other systems. Then we can really study and identify the optimal size. And this will be related to both penetration, accumulation, cell uptake, and so they are all connected in lymphatic distributions. Then this will be, in fact, the overall efficacies eventually.
So your work shows that you don't need to necessarily target it specifically and have no targeting moieties, but just pick the right size.
Yes, so
it could end up in the tumor. That's the bottom line, right?
right. So yeah, that's the first study of. So later we will evaluate some other active targeting ligands, see if we can further enhance the accumulation and the targeting effect.
No, but there's a lot of discussion in the literature and overall that do you really need, I mean,
right.
does active targeting really help or not, right?
We're studying one thing at a time.
Yeah.
Yes.
Now we're going to add the targeting moieties after we figure out
Right.
which size is involved.
But the point is you might not need a targeting moiety.
And we're going to compare.
Right. And then you can
without
compare, right?
targeting later.
Yes. Okay.
And are you, is this particle going to go through the NCL, or are you talking to them at all, or the characterization lab? Have you thought about that? Or?
Right. I think we're moving much faster than you.
Oh, then they so thanks to the animal models there.
Yes.
Yeah, so
Right.
internal collaborations will go, but that'll be.
It might be useful to at least have a presence there,
That's right.
right? To
And using
have some
different-
output of this work going there.
Right.
There might be some other value in that. Okay. Any
Okay.
questions? Great. Thank you.
Thank you.